DNA structure. But before we start, let us define first what is DNA, or the deoxyribonucleic acid. It is a genetic material that defines every cell or molecule known as nucleic acid, which consists of three domain components: the deoxyribosugar, phosphate, and nitrogenous base. Deoxyribosugar and phosphate serve as the backbone of the DNA, which can be seen here and here, while the nitrogenous base are bases that can be seen here and here. DNA is consists of two spiral nucleic acid chains that are twisted into double helix shape, just like in the figure shown. The twisting allows DNA to be more compact in order to fit within the nucleus and forms chromatin. Chromatin is a term for DNA as it packs into tightly coiled structure. During cell division, this chromatin condenses to form chromosomes. Prior to DNA replication, the chromatin loosens, giving cell replication machinery access to the DNA strands. And that will be the end for DNA structure. Steps in DNA replication. First step will be the replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. DNA has four different nucleotides or bases: the adenine, thymine, cytosine, and guanine. These different nucleotides form pairs between the two strands. Adenine only pairs with thymine, and cytosine only binds with guanine. In order to unwind DNA, this interaction between base pairs must be broken. And the breaking of these base pairs is performed by an enzyme called DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into Y shape, known as replication fork. Replication fork can be seen in the figure or in the photo. Replication fork is an area where the template for replication begins. However, replication fork is bidirectional. If it is oriented from 3' to 5' direction, then it is leading strand. While if it is oriented from 5' to 3' direction, then it is lagging strand. Step number 2, the primer binding. Once the DNA strand have been separated, a short piece of RNA called primer binds to 3' end of the strand. The primer binds will be the starting point for replication. Or in other words, in this step, this is where the replication begins. Step number 3, elongation, which involves DNA polymerases. DNA polymerases are enzymes responsible for creating new strand by a process called elongation. There are 5 types of DNA polymerase in bacteria and human cells. The polymerase 1, 2, 3, 4, and 5. Polymerase 3 is the main replication enzymes, while 1, 2, 4, and 5 are responsible for error checking and repair. While back in the process, DNA polymerase III binds to the strand at the site of the primer and begins adding new base pairs complementary to the strand during replication. In eukaryote cells, polymerase alpha, delta, and epsilon are primary polymerase involved in DNA replication. Because replication proceeds in 5' to 3' direction on the leading strand, and the newly formed strand is continuous. And in lagging strand, replication begins by binding with multiple primers. Each primers is only several bases apart, and DNA polymerases then add pieces of DNA, called Okazaki fragments, to the strands between primers. This process of replication is discontinuous. Then, the last step, or step 4, termination. Once both continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strand, and those primers that has been removed are replaced with appropriate base. And another exonuclease proofread the newly formed DNA to check, remove, and replace any errors. And lastly, the formation of phosphodiesters bond between the last and first nucleotide done by ligase will finally end the replication process. And that will be the end for DNA replication. Thank you for listening and watching.